Good morning, students. Today's topic is centrosomes and its functions. Centrosome is a microtubule organizing center, MTOC in short, of eukaryotic animal cell. It consists of two centrioles that are right angles to each other. They are present near the nucleus always and which are embedded in a mass of proteins. Centrosomes was discovered by uh, a person, scientist called as Benedin, and the name was coined by Boveri. They are found near the nucleus. Are centrosomes together. They constitute centrioles plus the centrosphere. What is a centrosphere? Centrosphere is nothing but a clear cytoplasm which is present inside that of the centrosome. Okay, then centrosomes are also called as diplosomes. Why they are called as diplosomes is because there are two centrioles in it, isn't it? Henceforth they are called as diplosomes. They're also called as diplosomes. Centriole structure. It is an array of 9 triplets of microtubules equally spaced around the perimeter. If you can see that, and the space between and immediately around the triplets is filled with a dense, amorphous, electron dense material, which you can see here, an electron dense material which is in the transverse veins, which is in red color, which you can see here. So this is the dense material which is between the triplets present there. Okay, so then they are arranged, or these veins are tilted so that they form an angle of roughly about 40 degrees to the radius of the cylinder. So if you can see this diagram very clearly, it shows, see, there are 9 triplets which are arranged around the centriole, which makes the centriole. This is one centriole and the other centriole, which are arranged right angles to each other, which are near the nucleus. If you see this nucleus here, and they are present here, so near the nucleus, and they have the 9 triplets which are arranged. If you can count, you can count 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, 9 triplets are arranged. Each are connected with a dense material, which can be seen here in this red color transverse veins. The centrioles are not surrounded by any membrane, but they are present in a matrix. Okay, centrioles have 9 plus 0 arrangement. What do you mean by that 9 plus 0 arrangement of microtubules? There are, as I told you before, if you can see in this picture, there are 9 microtubules which are present, 9 microtubule triplets are present there, and plus 0 arrangement meaning is what? Is that in between there is no microtubule present. Around the perimeter only you can see the 9 microtubules. Microtubules are made up of a tubulin protein. Microtubule have a substructure of A, B, C. If you can see that A, B, C is nothing but the first triplet, the second one, and the third one. If you can see a picture here, see, in this picture I have drawn the two consecutive triplets, isn't it? See here, the two consecutive triplets. I have named as A, B, and C. C is getting connected with the A, okay, which is making and transverse vein of 40-degree angle, which I told you, explained a little earlier. Then then the microtubule has 12 or 13 globular subunits. That's what which I have drawn here to you all, to show you all. See, if A, B, C is a triplet, one of the triplet which I have taken here and enlarged to make you show that how the 12 to 13 globular subunits which forms the which forms a one triplet there, okay, or the substructure A. Then out of which 3 or 4 are of these are shared with a B tubules, which in turn share several of its subunits with the C, which makes the three together, if they have to be together, isn't it? See, like this, if they have to be together, so few of those globular proteins are shared with each other so that they can interact with one another. Then next is this picture, which you can clearly shows you how the interacting has happened and how the cylinders are, the fibers are formed. Substrate A, B, and C. See the microtubule triplet, which you can see, and the centriole, and both the centrioles that you can see. I will come back to this picture little later. Linkers. See, what are these linkers now? A tubule of each triplet is linked with C tubule, which I have told you in my earlier small picture, isn't it? The C is getting connected with the A tubule. See, the triplet is linked with C tubule of neighboring triplet by a dense link, which I have told you a little earlier, and they are responsible for a radial. See, they are responsible for the see the a dense link, or they are responsible for a radial tilt, which I told you little earlier. They are the dense condensations from A tubule towards the center of the cylinder, forming a series of feet. Further at the proximal ends, the centrioles or the basal bodies occur connection between the tubules in a form of a hub spokes, or also called as cartwheel, which I will come back to this picture. See here, to this picture, if you can see here, the cartwheel connections and the hub in between, which you can see here very clearly. See, this is the hub which is there in between, and each of these are the spokes. See here, you can see the spokes which are present here. See, the spokes are present, and they are connected, and they have a feet here. This is the feet. Then see, the cartwheel consists of a 9 radial spokes, and these end in the dense feet. Okay, these end in the dense feet. That is what which is shown here in this diagram, and dense feet. Okay, then see here, you can see it very clearly. Spoke which has been labeled here. Okay, which is there here, and the central hub triplet microtubules are there, and C A to C linker, which is seen very clearly in this particular picture. Associated structures. What and all are the associated structures along with the centrioles? First of all, why did we study about the centrioles when we are talking about the centrosomes? See, centrosomes consist of the centrioles, isn't it? So henceforth, we are completely dealing with the structure of centrioles and its functions of the centrioles. Okay. So then, see, associated structures. These occur in the 9 amorphous plaques or the spheres of electron-dense materials which poorly defined outer limits near centrioles. So they are near the centrioles. There are few structures which are associated with the centrioles. These are called as macules, or it is a pericentriolar satellites. Centrioles are or can replicate themselves. How can they replicate themselves? Only when they have the DNA as a genetic material, correct? See, as the mitochondria and the cytoplasm do have their own DNA, isn't it? Same way, centrioles have the DNA and the RNA, and also they have certain kinds of proteins with them. Okay, so the double standard DNA is present in them, henceforth they can replicate themselves. Functions of the centrioles. See, they help actually in the formation of the spindle fibers during the cell division. This is a common function which you already know about it, correct? So the centrioles form the spindle fibers between the, I'll show you the picture here. See, these centrioles which are there here, which are forming the spindle fibers to join the centrosome of the chromosome, and they pull apart the chromosomes, isn't it? They divide the chromosomes into two halves. So then they and one more point I'll have to tell you, very important. See, if centrioles are there, they are going to help in the production of these spindle fibers, and they are going to help in the division of the cell. But some cells do not have the centrioles. There is an absence of centrioles. Then also the cell division takes place. Okay, that is what is a few articles says, and newly researched team has said that without the centrioles also the cell division can happen. They form the basal bodies. The centrosomes form the basal bodies, which give rise to cilia and flagella. This is a very important point that you'll all have to note down. See, the centrosomes or centrioles can give rise to the basal bodies, which in turn gives rise to a cilia or the flagella structure. What are the cilia and the flagella? Cilia is nothing but the locomotor function of the cell, isn't it? If it is a unicellular cell like paramecium or euglena, the flagella or the cilia is the locomotor structures for these two organisms. So, but when coming back to the animal cell, which cell do you require the cilia or the motion of mobilizing of the cell is happening? See here, the sperm, example sperm, a cell sperm when it is developing, if centrioles are absent in a particular cell, then the sperm cannot give rise to a cilia or a flagellum. This is a very important point. So a cell must have the centrioles if at all it is giving rise to a sperm. Okay, because it has to give rise to a cilia, which can which is going to facilitate for its locomotion to reach a target cell, that is egg. The placement of centrioles within the cell determines the position of the nucleus. So, where the centrioles are present, that determines the position of the nucleus. Henceforth, the centrioles are near to the nucleus, meaning the centrioles are in the center, little away from the center, so that they can place the nucleus right in the center of the cell. In turn, I'm telling you in that way, determines the position of the nucleus and the other organelles also. This is because the microtubules constitute the exoskeleton. See, the microtubules are going to form a complete mesh-like structure throughout the cell. See, like this. See the exoskeleton which I am showing you. The complete exoskeleton which you can see here, the microfilaments which are red in color. Okay, green in color is the microtubules. See, throughout the cell you can see the microtubules, and right in the center it is held by the mesh like structure. Okay, and blue markers are nothing but the nucleus there. Okay, so this is the exoskeleton which is being formed by the microtubules. So this is what is the functions of the centrioles and the centrosomes. Here ends up the topic. So any doubts you can ask me. Thank you. If you like the video, you can like it.